All right, let's dive into this. We're talking all about carbapenems today. It seems like you hear that name pop up more and more, especially when discussing those really tough infections to treat.
Oh, absolutely. And I think a lot of folks are interested in getting up to speed quickly.
Yeah.
I mean, even if they just know the name, there's a lot more to it than that. That's why we are doing this deep dive here.
Yeah, exactly. I'm really glad we are going to be talking about all this, together and all. So for someone who might just be learning about these drugs for the very first time, what are they all about? Like, what are carbapenems?
Carbapenems, okay, so they're part of this group of antibiotics called beta-lactams. That's like, the same family as penicillin.
Okay, right.
But carbapenems are like the big guns. They're really powerful and can tackle a really broad range of bacteria, even those that have gotten smart and resistant to other kind of weaker antibiotics. So
Hmm.
we often have to save these for those really serious infections when nothing else seems to be working. That's when they really come into play.
Yeah, that makes sense. They're sort of like the last line of defense.
Exactly.
Okay, so what gives these carbapenems that extra edge? What makes them, I guess you could say, so powerful and able to fight such a wide variety of bacteria?
Well, it comes down to their structure, how they're actually built. Now, they do have that beta-lactam ring, just like penicillins, right?
Okay.
But there's a key difference. It's all about this one carbon atom, right?
Okay.
In carbapenems, they got a carbon in a specific spot where penicillins have a sulfur. It's just a tiny difference, but man, it makes a world of
Oh,
difference.
wow.
And they also have a double bond in a certain part of their ring. These changes, they might seem small, but they make carbapenems a lot tougher. They can resist those pesky enzymes that bacteria use to, like chew up and deactivate a lot of the other antibiotics we have.
So they're built to last a little longer, huh?
They are. They are.
That's super interesting how that one tiny little difference can make such a huge, huge impact.
That's chemistry for you.
All right, so we know about the structure, but are there different types of carbapenems that doctors actually use? Does one size fit all, or is there a variety for different situations?
No, you're absolutely right. It's not just one type. We've got a few different carbapenems, and
Yeah.
each one has its own strengths and, little quirks. Imipenem was the first one on the scene.
The OG carbapenem.
Yeah, the pioneer. It's got a super broad reach, really effective against a wide variety of bacteria. But there's a catch. We have to give it with another drug called cilastatin.
Oh, okay.
Because imipenem, if it's given alone, it gets broken down too quickly in the kidneys by an enzyme there.
Ah, I see
So
that.
cilastatin basically protects it.
Hmm.
Now, we have meropenem. It's similar to imipenem in its wide range of activity, but we don't need that cilastatin sidekick. It's good to go on its own, and plus it carries a lower risk of causing seizures, which is a big plus.
Yeah.
Then
Yeah.
there's ertapenem. Ertapenem's a bit more specialized. Its spectrum of activity is a bit narrower, meaning it doesn't cover as many different types of bacteria.
Gotcha.
Like, it might not be as effective against certain troublemakers like Pseudomonas and Acinetobacter, but it has a really nice long half-life. You can give it just once a day,
Yeah.
making it really convenient for patients.
That's pretty huge.
And finally, we have Dorepenem. It's got that broad activity similar to Meropenem, and it's very stable when we prepare it in those oddity solutions.
Ah, that makes things easier in a hospital setting.
It definitely does.
Okay, so we've got a whole toolbox of carbapenems, each one for a different job. But I'm curious, how do they actually work? What's their secret weapon against these bacteria?
Well, like all beta-lactams, they mess with the bacteria's ability to build their cell walls. Basically, they attack their construction crew.
Clever.
They bind to these specific proteins called penicillin-binding proteins, or PBPs for short.
PVPs, got it.
And these PBPs are really crucial for building a strong and rigid cell wall, which bacteria need to survive. So when a carbapenem latches onto a PBP, it's like throwing a wrench into the machinery. The bacteria can't build their walls properly, and, well, they basically fall apart and die.
And the reason carbapenems are so good at this, against so many different bacteria, is that they can get through the outer layers of those tough gram-negative bacteria.
You got it.
They can get through those channels, the porins.
Exactly. And not only that, they can bind to different types of those PBPs, making them very versatile.
So they're like master key. Huh.
Yeah, sort of.
Okay, so they're clearly very effective. But we've talked about their broad activity. Can you give some specific examples of like what kinds of infections carbapenems are typically used for?
Oh, yeah, sure. So we're talking about the serious stuff here, like really serious. Hospital-acquired pneumonia, especially those that develop while someone's on a ventilator.
Oh, wow.
And then we've got those complicated intra-abdominal infections, like when there's a perforation or an abscess. And complicated UTIs, urinary tract infections that is spread beyond the bladder.
Yep.
And then there's bacterial meningitis, which is an infection of the lining of the brain and spinal cord.
Yeah.
And that's where meropenem really shines, because it can actually cross the blood-brain barrier.
Oh, that's incredible.
Then, in folks with weakened immune systems, like those undergoing chemotherapy, and of course we use them for sepsis, which is a really dangerous whole-body inflammatory response to infection. These are all cases where other antibiotics often just don't cut it.
Wow, it's like a whole team of specialized superheroes,
They kind of are.
each with their own strengths. Okay, so we've talked about how important these drugs are, their strengths, but there's this big elephant in the room, right? Antibiotic resistance. We hear about it everywhere. Are bacteria starting to become resistant to carbapenems too?
Unfortunately, yeah.
Yeah.
It's happening, and it's a major worry. The biggest threat we're facing is the emergence of these enzymes called carbapenems. And these are like the ultimate weapon for bacteria.
Oh no.
They can actually break down the carbapenem molecule, rendering it useless.
So they just eat up the antibiotic.
Yeah, essentially. And what makes it even scarier is that the genes for these carbapenems can be shared between bacteria, like trading secrets.
Oh, that's not good.
They do this through these little packages of DNA called plasmids. Think of them like little flash drives that bacteria can pass back and forth.
Oh wow, so resistance can spread like wildfire then.
Exactly. And some of the most notorious carbapenems, the ones causing the biggest headaches,
Right.
include KBC, that's Klebsiella pneumonia, carbapenem,
Okay.
and NDM, New Delhi metallo-beta-lactamase. There are others too, like VIM, IMP, OXA48 types, the old rogues gallery.
And are those plasmids, are they specific to one type of bacteria or can they jump between different species?
Well, that's the scary part. They can sometimes jump between different species. So resistance that develops in one type of bacteria can potentially spread to others.
So it's not just containing it within one species. You might have to worry about it spreading to others.
Exactly.
Okay, so that's a little frightening.
It is.
So are there any other ways besides these super enzymes that bacteria can develop resistance to carbapenems?
Yeah, there are a few other tricks up their sleeve, though they're not as common. Remember those porins we talked about? Those channels that let the carbapenems into the bacteria?
Yeah.
Well, sometimes bacteria can change these porins, making them less permeable. So the carbapenems have a harder time getting in. For example, changes in a specific porin called OPRD in Pseudomonas aeruginosa can make it really resistant to imipenem.
So they just shut the door on the antibiotic.
They do. Another thing they can do is use what are called efflux pumps. These are like little pumps in their membranes that just spit the drug back out as fast as it gets in.
They are not messing around.
They are not. And then sometimes, though it's less frequent, the PBPs themselves can mutate. So they change shape a bit, and the carbapenem just doesn't bind to them as effectively anymore.
So it's like they're changing the locks on the door.
Exactly.
Okay, so it's a constant arms race. Evolution in action. Bacteria are adapting. It's a little intense. So I'm curious, where do these carbapenems even come from in the first place? Like, where do we get them?
It's actually a pretty neat story. The original building blocks for these drugs were discovered in nature.
Oh, really?
Yep. They're produced by soil bacteria called streptomyces. Those guys are responsible for a lot of our antibiotics.
No, right, of course.
So they produce these molecules like theanomycin, which are kind of like the precursors to carbapenems. But those natural versions aren't quite ready for prime time in medicine.
Oh.
Scientists take those molecules and tinker with them in the lab, tweak their structures. That's how we end up with the carbapenems we use in the clinic, like imipenem and meropenem.
So from the soil to the lab to the hospital, it's pretty amazing. It really underlines how important these drugs are, but also how fragile that whole system is. We're relying on nature and our ingenuity to stay one step ahead of these superbugs.
Absolutely. This whole deep dive, it really highlights the ongoing need for new research and new ways to combat infections. And it really underscores how crucial it is that we use antibiotics responsibly. We can't take these drugs for granted.
No, absolutely not. This has been so informative. I feel like I have a much clearer picture of carbapenems now, why they're so important and the challenges we're facing. I think the big question everyone's left with after this is, given how quickly bacteria adapt, what other strategies, besides just new drugs, might we need to think about to really win this fight against infectious diseases?
That's the million-dollar question.
It is.
It really makes you think.
It really does. Okay, well, this has been a fantastic discussion. Thanks so much for walking us through all of this.
You're very welcome. It was my pleasure.